Good morning, everyone. Welcome. Welcome to the Garvan Institute of Medical Research. It is fantastic to see so many of you here in the audience. I believe we also have around 200 people joining us via Zoom and Facebook Live from around the country, which is just fabulous. Thank you so much for being here. Our public seminar series is a really fantastic way to learn about the very latest that's happening in Garvan's research and to hear directly from the incredible researchers who are doing the work. So thank you so much for being here today, for your engagement, your interest, and your support. Now, for those of us who might be joining us for the first time, my name is Dr. Vivian Richter, and I head up the marketing and communications team here at the Garvan Institute. I've been at Garvan for more than 6 years and have had the privilege of witnessing some incredible transformations in how we understand and treat disease. It really is such an incredibly exciting time in medical research. Before we kick off today, I would like to acknowledge the traditional custodians of the lands on which we meet today, the Gadigal and Bidjigal people of the Eora Nation. We pay respects to elders, past and present, and recognise their continuing connection to this land. Now, as a bit of housekeeping to start, we will have a Q&A session at the end of the seminar, and I encourage both our guests in the auditorium today and our audience online to ask questions. For anyone who's joining us online, please feel free at any point during the seminar to type in your question into the Q&A function that's at the bottom of your screen. And for those of us in the room today, please feel free to raise your hand during the Q&A session at the end, and a Garvan staff member will run over a microphone to you. Our bathrooms, critically, are just at the bottom here, outside the auditorium on the ground level, and if you haven't done so, please do switch off your mobile phone or put it on silent. Today's topic is pancreatic cancer, a disease that is incredibly difficult to treat and often very difficult to diagnose. The pancreas is located very deep within the abdomen, and by the time most patients present with symptoms, the disease is already spread to other organs. Now, with the current 5-year survival rates of less than 13%, there is no doubt that there is an urgent need for more effective treatments. And today you're going to be hearing from two world leaders in pancreatic cancer and cancer research, Professor Marina Pajic and Professor Anthony Joshua, who are the co-directors of the Translational Oncology program here at Garvan and who are leading transformative research projects that are set to really shift the dial on this disease. We will also hear from Daniel Goulburn, who has devastating experience with pancreatic cancer himself and whose advocacy has been instrumental in changing public awareness. Thank you so much, Daniel, for being here. Now, it is my privilege to welcome our first speaker today, Professor Marina Pajic. Please come on up to the stage. Professor Pajic joined Garvan in 2013 and has developed a research program that leverages cutting-edge technologies, patient-derived models, genomic data, extensive clinical partnerships to really advance research in this area and change how we understand the cancer's complexity and ability to treat. Welcome, Marina.
Thanks very much, Vivian, for that lovely introduction, and thank you all for being here today. It is a really great pleasure to be able to hopefully share some exciting new research that's happening within our program and also in our laboratory today. With that, yes, okay, we're on it. Well, as Vivian already mentioned a little bit to you, it is important before we start talking about pancreatic cancer, we need to really understand and know the role of the pancreas itself. So it is an interesting organ. It is sort of tucked away behind the stomach in our body. And it is actually connected to a number of vital organs that have very important functions in their own right. Two main functions of the pancreas, in particular, we need to highlight, are its endocrine role. So pancreas has some specialized cells within, there are specialized cells within the organ that produce specific hormones. So these are chemicals that regulate blood sugars, and probably one of the best known is insulin in this context. The other role of the pancreas is its exocrine role. So in addition, so to producing hormones, different cells within the organ also produce enzymes that help break down and digest the food. So things like amylase that breaks down carbohydrates, protease that breaks down proteins into smaller components, and lipase that deals with fats. So, you know, although it's a sort of a hidden organ, it is actually connected to a whole bunch of important things that make our bodies run efficiently. And so when things go horribly wrong, I guess one of the big examples being pancreatic cancer, the effects and the impact of this is quite significant. And so again, just not really to dwell on this too much, but just to touch on some of the dismal statistics surrounding the disease. Survival rates have actually improved for pancreatic cancer in the last decade or so. So they were actually considerably lower. So we were looking at 8%, and we're now looking at 13%, 5-year survival rates. Still, and this is due to, mostly due to introduction of some of the polychemotherapy regimens in the context of treatment of the disease. However, there's a whole lot more that we can do in this space, and that is really the goal of our lab. It's to try to develop new and more effective treatment approaches for patients with this disease. Unfortunately, as it stands, so the disease is actually predicted by 2030 to become the second leading cause of cancer deaths. So it is a really important area of focus for us and numerous other labs around the world. Okay, so this is really just to illustrate to you again the difference between what the tumor looks like. So this is just histology, cutting through a tumor that's been collected and then sectioning it and pathologically looking at the section itself. So you can see, in contrast to the normal pancreas on the left, that looks very organized, although you may not be specialists in this area and really understand all the specific structures. But here we have, so I can just highlight a few. We have, if this works. Does it? No? Can we get it to work? So on the right, we have these little cells. I shouldn't move away from the microphone. No? No. We have islets, which contain these specialized cells. So these are the cells that make the hormones that I mentioned earlier. And then we have the other cells that are secretory acids. So these are the cells that actually produce the digestive enzymes that help digest our food. But really, in contrast to the section on the left, you can see that the section on the right, which is the cancer, it's actually very disorganized. So it's a completely different looking tissue. And so that's really what we're dealing with here. And although, as Viv also mentioned before, our lab has spent a lot of effort in the last 10 years or so studying the genetics of the disease, and we're still very much interested in understanding how different mutations that cause these cancers to arise in the first place and then enable them to progress, develop resistance on treatment. What I want to talk about today really is the role that the environment plays in how it actually talks to the cancer cells. So these are the cells that surround the cancer cells as they grow and develop, and they further produce different stimuli that help these cancer cells grow. So today the focus will really be on the role and also perhaps highlighting some of the interesting ways in which we can manipulate cells within this environment to then block some of this pro-tumorigenic, pro-oncogenic signaling to improve response to other types of agents that are already used in the treatment of pancreatic cancer. So just the image again on the right in purple, so the purple dots really highlight the tumor cells themselves. So what you can see here again in the section and also illustrated in schematic here, on the left is that there is this section highlighted in red. So these are these what we call stromal cells. So these are traditionally normal cells that are present in our body that are here being corrupted by the cancer cells. They surround the cancer environment, and then they produce these various stimuli that help cancer cells grow. Another important component of this stromal environment that I'll go into detail a little bit more is the ability of some of the cells that are present in this environment. They're called fibroblasts. They're also, again, illustrated in this schematic. So fibroblasts are cells that are normally present, for example, in our skin. They make collagen. Collagen is one of the, buzzwords. I mean, it's been a buzzword for the last, 5 to 10 years now. They're very important cells. They make lovely collagen, make our skin look younger. Unfortunately, here what happens is that these cancer cells can corrupt the otherwise normal function of these fibroblasts and then make them produce excessive collagen. So it's this excessive matrix that then creates this dense-like, almost fibrotic scar-like tissue that surrounds, envelops the cancer cells, produces signals that help the cancer cells grow further, but then also creates this physical barrier to therapy entry. And so today I'll just highlight some of the results that we have actually in this space in terms of our ability to block some of these signals, specifically in this fibrotic area. And one of the key proteins or molecules that we've been studying for a reasonably long time now is this protein called ROCK, Rho-associated kinase, which, again, just highlighting here, it actually plays a number of different roles. We've shown that it's highly present in the pancreatic cancer cells. We've published that previously. And what we also know about this molecule is that it drives production of this fibrotic scar-like tissue, among other roles. So it also plays a role in how these cancer cells may move from the primary tumor to the metastatic sites, and it also plays an interesting role in therapy resistance. Today, so we also actually, as part of the broader translational oncology program and some of the work of our collaborators, we have 2 new interesting antifibrotic therapies that are hopefully towards the end of this year entering early phase clinical trials. But today, what I want to focus about specifically is this compound called RXC007 or zelasudil. And this is the protein that selectively blocks ROCK signaling. So just showing you a few key highlights from our already published work, what we've just to kind of sort of set the scene. So this shows you, again, a number of sections. So we've analyzed using automated imaging analysis. We've looked at collagen levels, so this fibrosis levels inside pancreatic tumors, inside a cohort of 231 samples. And so that's highlighted here with this purple imaging. So the purple signal shows how much of this collagen is present inside these tumors. And what we've basically shown is that a high proportion of these patient tumors have medium or so detectable or high levels of fibrosis present inside. So it's clearly an important component of the surrounding environment of these cancers. And then looking at our models, we were also able to show that these models recapitulate some of these key features that we see in the patient tumors. So these are animal patient-derived tumor models of cancer that we have established over a number of years here in our lab. So on the left, I'm just showing you the sort of control images. So these are tumors that have just grown. And so again, either on the top panel using the purple, so this is just fluorescence, collagen fluorescence under the microscope. We can pick up these signals. On the left, we're using a more traditional stain that's used clinically, which is called Sirius red stain, and labels up collagen so we can see and detect these levels of collagen inside the tumors. What's also really interesting is that when you treat these tumors with chemotherapy, so that's really highlighted on the right side, either with one of the standard of care therapies called gemcitabine, so highlighted there just as gem, or below FOLFIRINOX, which is a polychemotherapy that's used in the clinical treatment of these patients, we can see that there's a dramatic increase in the level of this collagen inside these tumors. And it kind of, I guess, it is logical. It makes sense in the sense that these tumors are obviously trying to protect themselves in some way from the treatment itself. And we can quantify this. So again, so this is all automated. It's usually using computational tools to scan, analyze these images, quantify signals, and then we can use statistical methods to show that, you know, something is biologically or statistically increased inside these tumors. So what happens when you try to block ROCK signaling in these models? So again, this is just showing you some published data. We've published it a while ago now. In the first instance, using a dirty, so we call it dirty, it's not very selective, and it's not particularly clinically applicable inhibitor. So it's a small molecule compound that blocks this pathway. It's called Fasudil. And so what we've shown here, and really what you want to see here on the bottom, so this is called a survival plot. These are, again, our animal models of advanced metastatic pancreatic cancer. You can see the black line dropping basically from 100% survival. So the animals quickly reach ethical endpoint, at which point we need to euthanize the animals and we collect the survival data. So you can see the models are very aggressive, and you can also see the green line, although it shifts slightly towards the right, the chemotherapy, in this context, gemcitabine Abraxane, so highlighted there in green, it improves the survival, but not particularly dramatically. What's really interesting is that when we use, and that's highlighted by the blue line, which shifts further, even further to the right now, is that when you use the ROCK inhibitor in combination with the chemotherapy, we're now starting to see more dramatic increases in the survival of these mice. So that was the first piece of, I guess, promising data that we were able to get with a dirty form of this inhibitor a few years ago now. So that has since led to an interesting new collaboration to try to assess some of the more selective next generation ROCK inhibitors. So that's RXC007. And the way it also came about is really meeting up at an international conference with some of the members of this pharmaceutical company. It's called Redx Pharma. It's a UK-based company. They had this interesting compound that has been undergoing, firstly, preclinical development in their laboratories, and then has since proceeded to clinical development in the context, so in non-cancer conditions. So they were looking at here, specifically using it for idiopathic pulmonary fibrosis. So this is lung fibrosis. It's another chronic and low survival disease. And so what I'm highlighting here is really just some data from their models of lung fibrosis. So really highlighting that when you use the ROCK inhibitor, so that's RXC007, and now you're all expert in histology analysis. So this is all very obvious to all of you. I'm highlighting 4 different images on the bottom, and then also quantified data. So first we have control lung, so that's a healthy lung. Lung obviously looks different from the pancreas or the pancreatic cancer, but what you want to see is actually a whole lot of these holes, because this is where oxygen comes through, and these are your alveoli structures. So that's sort of a normal lung. What they call their allogeneic control, this is their actual model of lung fibrosis. There is now an increase in this red signal, and there's a lot less of these holes within. So you want thin alveoli alveolar structures. But what's actually important is that whether you use a low dose inhibitor or high dose of this inhibitor, you now start to revert this condition, and you again see better, I guess, a lung that now looks more like a healthy lung control. And since then, they've also been granted special drug designation by the FDA to investigate this compound clinically for the use in IPF, and it's currently in phase 2 trials in IPF. And so we were obviously very interested in collaborating with these guys. They're also very lovely and very friendly and share all of their information, which is beneficial when you're talking to pharmaceutical companies. And so what we were able to do is then apply their compound and test it in our models. So I'm just showing you a snippet of data here. So here we took, firstly, we wanted to see whether these compounds can reduce fibrosis in our models. And so we treated with RXC007, so that's just highlighted there with the ROCK2 as ROCK2 inhibitor. Again, so using the standard Sirius red stain, so this is the collagen stain, what we saw is a reduction in this collagen coverage. We can then also confirm this using the collagen fluorescence as an imaging tool as well. And again, what we're comparing is the right panel versus the left panel. We saw a drop in the collagen signal. Now, what I've mentioned before is also that these cancer-associated fibroblasts, so these cells, so there are specific specialized cells that produce this matrix. So then what you can also do, we have specific markers that can mark up these cancer-associated fibroblasts in situ, inside these tumors. So just using two protein marks, in this context in red, alpha-smooth muscle actin, and then PDPN or podoplanin in green, we can again use these automated imaging tools. They scan the sections and then they look for cells that contain both of these signals. And then we can quantify the number of those cells that are present inside these tumor sections, and then we can compare what the treatment does to the control. And so it was, I guess, nice to see that associated with this reduction in the fibrosis on the left, what we saw is the reduction in the activation or presence of these cancer-associated fibroblasts inside these tumors. So it just signaled, so those are sort of initial signals that the drugs are doing what they're supposed to be doing. Or I guess what it's doing in the context of lung fibrosis, it seems to be having a similar effect in the context of the pancreatic cancer fibrosis. And so just, I guess, highlighting some of the things we can actually do in the lab as well, which is really nice, using computational modeling. Again, we can mask. So what you can do is you can create masks of this red stain. So that's just highlighted on the right. And then you can study a whole heap of other effects. So we can, it's just basically giving us additional supportive data to show that, yes, we now have a less stiff, less organized fibrotic matrix that envelops these cancers. So we see, firstly, in the middle, we can study the length of fibers of these collagen fibers that are present inside these tumors. So we see a reduction in the length of these fibers. And then we can also see, we can look at the difference in terms of the complexity of the fibers and how they mesh with each other. So again, we see a decrease in this in the overall mesh structure inside these tumors. Ultimately, we want to see what happens to the survival of these animals or animal models of cancer. So the first way we also looked at this is using a more rapid model. So this is where we inject cells into the spleen, and then we study how these cells spread into the liver. So as I mentioned before, metastasis or cancer spread is a key feature of pancreatic cancer. And so this is just one way to look at how our drugs might affect the spread of the cells when they leave the primary tumor and how they spread into the liver. And so what I'm highlighting here, so these are now liver sections. Again, you don't really need to focus, I guess, but you can see that there is this organized structure. So there's a lot of red staining, and then it's, I don't know, even, so quite interestingly, my 9-year-old daughter has been counting the metastases for me quite successfully. You can see these changes in the patterns. So the metastases are these darker blue, darker purple. I wish my thing worked, then I could actually highlight. But there you can see them all over the vehicle controls, and then we see a reduction in both the size and also the number of these metastatic foci that we can detect and count in through these liver sections. So that's one readout of efficacy. The second readout involves actually testing this in quite a number of different models. Here I'm just highlighting one of our advanced models of metastatic disease. So what we're, again, you're all experts at looking at survival curves now, but really what we're looking for is the comparison of the purple, which is the chemotherapy. So in the top context, so it's gemcitabine Abraxane chemotherapy, again, one of the standard of care treatments. We now see whether we use, and here we tested two different treatment strategies using the ROCK2 inhibitor, a more chronic treatment, and then a more episodic treatment. So here what we're really playing with is looking at the amount of ROCK or the targeted therapy that we need to give in order to improve survival of chemotherapy agent. Because understandably, patients who present with pancreatic cancer often present very sick. So we do have to be mindful when designing combination treatment strategies, that these are, I guess, at least reasonably feasible when applied in a clinic setting. So regardless, though, what we saw here is a significant increase in the survival in this context, in this model, and then also even more dramatically when we look at a model. So this is, again, another highly fibrotic patient-derived tumor model of pancreatic cancer that's resistant to FOLFIRINOX polychemotherapy. So that's really the purple line really overlaps with the black, which is the control line. So the chemotherapy really doesn't work in this setting. When we apply the ROCK2 inhibitor, we now are starting to dramatically shift survival in this animal model. And so that really brings me, I guess we are a preclinical lab, and so we do want to bring you insight around this disease, but ultimately the goal is to try to translate these findings into clinical trials. And so this really is about increasing access to new agents for Australian patients with pancreatic cancer. And so I'm very, very excited actually to be able to tell you that we do have two early phase trials, specifically in the context of these antifibrotic therapies. Here I'm just showing you a clinical setup for the Zelasutil compound, RXC007, which we're hoping to open later this year. But Anthony can tell you a lot more about this because he's the clinician who's actually leading these trials. But really what I'm showing you here is that, this will be a two-part study. On the left, we are looking at, because although Zelasutil has been shown to be safe as monotherapy in non-oncology settings, it hasn't been combined with chemotherapy. So it still needs to undergo this dose escalation phase before it can be expanded into the sort of expansion cohort. And really what we're interested in is determining, firstly, the safety and tolerability of the combination, and then starting to look for signals of efficacy. And of course, we're very much interested in all the correlative science, the deep science that we can then do on the patient material that we can collect as part of these studies. So that's later on this year, but I will now open it up and invite Professor Anthony Joshua to come to the stage, who is the co-director of the Translational Oncology Program with me at the Garvan, and also head of phase 1 oncology at St. Vincent's, as well as PI of the New South Wales Early Clinical Trials Network.
Thank you.
Welcome, Anthony.
Thank you very much, Marina, and thanks for the organisers for having me speak and highlight some of the work that we're doing. Granted, the information that my talk is a bit more, I'm a bit more of a sledgehammer. Marina is a bit more precise and scientific. So the nature of what I'll be telling you is a bit more, demonstrating a bit more of the angst that we have in the clinic, trying to get medications to patients as soon as possible. And so it's a slightly different approach, but it certainly complements what Marina has talked to you about. So one of the things that we're trying to do in the clinic is to accelerate the pace of drug development. So typically, when you find and you develop a new drug, one of the ways you can accelerate that is to repurpose a drug which already exists rather than creating a new molecule. So creating a new molecule is certainly, well, if you own it, far more financially feasible, but if you're passionate about helping patients, sometimes it's faster to take an existing drug and say, you know what, this drug may work better in this setting or in a certain a way to give it with something else. So similar to what Marina has told you, taking a drug from a company that was developing it and had already safety data in lung fibrosis, and then saying, you know what, that actually that fibrosis is also important for pancreas cancer is an example of that. And so you can see here how long it takes to develop a drug. If you add up all those years, we're looking at 10 plus years to take a drug from the clinic all the way into human beings and to get some results where you can actually then make it available for Australians. Here's my pointer. So this can take quite some time. But if you take a drug which is already in phase 1 or phase 2, we can cut this down in the order of sort of nearly 7 years if we eliminate this component, because that's done already. And we've come in with a discovery that can take the drug which already exists and repurpose it. So what is that drug? So when I first came to the Garvan, I had a few discussions with Marina about this strategy about drug repurposing. And there was a report back in 2016 about a patient who was in, I think, Texas, who took this drug called itraconazole. So itraconazole is a drug that human beings take when they have a certain type of fungal infection. People that get certain types of blood infections can take this drug. It's an antibiotic, but antibiotic against funguses. So people that have thrush sometimes take it, for example. So this patient had pancreas cancer. They couldn't cut it out. So what they did is they gave him some chemotherapy and gave radiation at the same time to shrink it. Unfortunately, it grew shortly after that. And then they gave this chemotherapy we don't give anymore, which is gemcitabine, which is the chemotherapy, and another drug called alotinib, 3 times, and that didn't really make a difference. The patient then caught this infection called histoplasmosis, which is a type of fungal pneumonia you can get in Texas, and then took this drug, I think, in the hospice to get over this fungal pneumonia that they had. And then the tumor shrunk enough on that drug, they could then have it cut out. And so it was a pretty amazing response, a case report that told me that actually maybe there was something going on here. So we looked in literature, and in fact, there's a lot of things that this drug itraconazole does that may speak to the fact and explain why it worked. So it's a drug which people have then, for various reasons, have said, you know what, it may have anti-cancer activity, and what could this anti-cancer activity be? So it does a number of things. To give you an example, one of the things it does is it acts cholesterol in your blood. This acts as an anti-cholesterol. So cholesterol happens to be important in cancer as well, because the cells need cholesterol, the cancer cells need cholesterol to just live. And so if you're taking something which is a reverse image of cholesterol, you can block the cholesterol in the cancer cell. And that's particularly important in a pathway here called hedgehog. So hedgehog is a growth pathway in cells, and it makes certain cancers grow, including pancreas cancer, but also makes some people may have little skin cancers called basal cell cancers on their skin grow. And there are certain people that have a lot of basal cell cancers on their skin because of genetic reasons, and those people can take itraconazole to stop those skin cancers growing. So we know that's, and I use that in my clinic, in my skin cancer clinic, but we know that it has other things by blocking some of the pathways in the cell, either directly or indirectly related to the cholesterol. It can do things like stop blood vessels growing around cancers, denying them food and denying them oxygen. It can stop the cell dividing, and it can also cause the cell to sort of commit suicide. So it seems to have a lot of applications and has been tested in a number of clinical trials. We know that it can, for example, help prostate cancer a little bit, and it's been tested. It can potentially help breast cancer. I certainly told you in the clinic it's sometimes used for basal cell cancers. So it's got this wide applicability in cancers. And so part of the question that my lab asked was, is there anything we can do to make it work better? So let me just take you through this. I know it's a lot of information on the slide, but I'll take you through it one thing at a time. So the initial thing we did is we took some cells and we treated half the cells with this antibiotic, and we treated half the cells with nothing. So basically we grew them up, and then what we put into these cells was an entire library of viruses. Each virus contained one gene from the human genome, and so that gene was taken into the cells, and because of this technology called CRISPR, which you may have heard about in the press, we were able to knock out every single gene in the human DNA in millions and millions of cells in these flasks. So, and then we said, well, what genes differentially disappeared if the cells were treated with this itraconazole compared to cells that weren't treated with this itraconazole? And so we came up with a here, and you can see here are the two cell lines that happen to be ovarian cancer, but that was not the point. And we came up with the sort of a list of cells, a list of genes which were selectively lethal in the presence of the itraconazole. So telling us that there is a weakness. So in the presence of this drug, this itraconazole, this antibiotics, you can exacerbate the cell killing by knocking out extra genes. And so when we made a list of those genes, we realized that actually a lot of those genes had to do with the cell probably trying to survive by recycling the garbage in the cell in an organelle called a lysosome. So in brief, the cell gets under stress, it realizes it's under stress, and so it ramps up its recycling mechanism to keep feeding itself. But that gave us a clue that if we could block that recycling mechanism, we could kill more cells. And how do we block that recycling mechanism? So fortunately, it appears that the recycling mechanism is the recycling bin in the lysosome is very acidic. That's part of the reason why it degrades stuff, which then gets fed out of the recycling garbage bin and goes back into the cell to be repurposed. So if you turn the acid off inside the recycling bin doesn't work anymore. And so fortunately, there's a drug which is already available that does that. That drug is a drug called hydroxychloroquine, which is used for malaria treatment and has also been used for various cancers because people had some idea that that might help certain cancers. So what we do in this experiment, we then said, well, look, most of these genes in this list are in that recycling bin. So if we turn off the recycling bin with this drug, maybe we can kill more cells. And in fact, in the cell line experiments, you can see here that as the concentration of this itraconazole drug goes up and the concentration of this chloroquine, hydroxychloroquine drug goes up, you can get what we call synergy. So that's 2 + 2 = 5. So you kill a few cells with one drug, you kill a few cells with the other drug, but this experiment tells us you put the two drugs together, you get extra bang for your buck. Your 2 + 2 = 5, and that's what all the red is. So all the red is the drug's working better than we would otherwise expect in the bottom right-hand corner of every little box. So that was quite seminal, and we published on this work in 2022 to say, this is an excellent strategy, no one's ever thought of it before. Can we treat human beings with both of these drugs, which you can just pick up from the local pharmacy, to help kill their cancers? And so the first cancer that we did it with was ovarian cancer, and we found that actually this CBR means a clinical benefit rate. So how many women who had ovarian cancer that it was otherwise effectively untreatable, they weren't suitable for other treatments, could benefit from this drug? And the answer was about 50%. And this is these months along the bottom, 1, 2, 3, 4, 5, 6, 7 months. So, and the black is the patient stopping treatment. But you can see that what we were encouraged by the fact that with a simple strategy, which we discovered in the lab, we were able to help women control their ovarian cancer for sort of 5, 6 months without experiencing really too much toxicity. As I said, these drugs are available in the local pharmacy, not expensive, easy to take, and The green box here in this lady had the tumour shrink visibly on the CAT scan, which I'll show you in a minute. So that was a very encouraging discovery for us. And this is a picture of, and you can see along here as we measured one of these deposits, it shrunk from 13 by 5 millimetres down to 9 by 6 millimetres, and that's that one. That's the one up here, which is sort of shrunk, and you can see that's smaller over there. So that was a very important proof of concept for us that suggested that actually this laboratory discovery actually had real clinical implications. And so what we're doing now is we're trying to put those two drugs together so that instead of having to juggle taking two drugs, we can tweak the chemical composition of a fused drug to make it work even better. I can't put that in a slide because that's secret, but I'm telling you anyway. The other aspect, which after discussions with Marina, we were even more excited about, was making the itraconazole work better by activating the immune system. So what Marina has discovered, and that she didn't highlight as much today, but this itraconazole does have incredible effects on a part of the immune system called the macrophage system. So the macrophages are sort of helper cells that help your T cells, which are the anti-cancer part of your immune system, attack the cancer. They provide the supporting infrastructure, if you like. They're the foundations of this attack when you get an immune system attacking cancer. And there's two types of macrophages. There's the M1 macrophages, that's the ones we like, which make, which digest dead cancers and help the T cells come in. And there's M2 macrophages, which we don't like, which are the ones that partly contribute to the fibrosis that Marina talked about and make, are used in wound repair. So you can imagine that that's probably a bad thing, to get more blood vessels into cancer, to help put more fibrosis into cancer. And so Marina found, and this is Marina's work, that giving mice with pancreas cancer a little bit of itraconazole when they have other drugs here which turn on their immune system in two different ways. So just briefly, this PD-1 drug turns on the immune system just around the cancer, and this CTLA-4 drug turns on the immune system as a master switch. And many of you who have friends who've experienced cancer would have had they would have had some of these drugs in the clinic. That's what the Nobel Prize was given for the two men that discovered these drugs. But you can see that in terms of surviving mice, the mice lived longer, men and mice, the mice lived longer if they had the itraconazole together with the immune stimulating drugs, because the immune stimulating drugs don't work on those macrophages. So something has to get those macrophages to switch from those nasty ones that make the fibrosis and bring in extra blood vessels to macrophages which help support an attack on the cancer. And that's what we hope the itraconazole is assisting with. And Marina's work has demonstrated that's certainly the case in these mice, which with other slides, which I won't bore you with. So we've now, again, translated some of these discoveries with our pharmaceutical partners into the clinic, and in a sort of exciting paradigm-shifting approach, have said what we're going to do is we're going to give these drugs that turn on the immune system to patients who have been treated for their pancreas cancer, but in whom the pancreas cancer has not yet broken through the chemotherapy. Because you can imagine the chemotherapy is quite toxic, and what we're looking for is now this sweet spot when patients have an intact immune system and are well enough that we can turn on the immune system to help attack the pancreas cancer, but they're finished with the chemotherapy. So no one's done that before in pancreas cancer. So we hope that we've found the sweet spot where there's enough get up and go in the immune system that we can attack the pancreas cancer with this triplet combination and have some meaningful effect on the pancreas cancer by adding in this extra drug, which will hopefully change the way the immune system attacks the cancer in a way that pancreas cancers haven't seen before. So it's very exciting work, and it is just been submitted to the ethics committee to get that set up just last week. So we're well underway in terms of getting that into the clinic. So just to summarize then, I think the work that Marina and I have discuss with you today have shown that we're certainly on the path to developing druggable targets for patients with pancreas cancer. And those targets are not just the cancer itself, but also the supporting stroma, that fibrosis around the cancer that stops the immune system getting in, stops the chemotherapy getting in. And also adding to that, an attack with the immune system. So we're bringing in this third pillar into the tumour microenvironment, angry immune cells, to help also attack the pancreas cancer. So thank you very much for your time. I'll now hand over to Vivian.
We now have the great privilege of hearing from Daniel Goulburn. Daniel lost his wife to pancreatic cancer in 2014, and after gaining an understanding of the truly dismal outcomes, the heartbreaking statistics, he co-founded Purple Our World, which is a global movement to raise awareness. He also works very closely with the researchers in this building as a patient advocate. The incredible work of our consumer advocates means that a patient perspective is considered throughout the research pipeline. I'd also like to acknowledge one of the other consumer advocates who work really closely with Garvan researchers, Jan Mumford, who's joining us online and who has just acknowledged Marina's presentation. Thank you so much, Jan, for joining us today. Daniel, your advocacy continues to push for earlier detection, better outcomes for patients with this disease. Thank you so much for joining us today and for sharing your perspective. I'd love to start our questions today with a question around Rochelle. Could you tell us about her?
Yes, thank you, and thank you very much for Garvan for inviting me to be here this morning. Rochelle was 57 when she passed away in 2014. She was diagnosed in March of 2013. We were considered lucky because we had 16 months between diagnosis and death. Rochelle was a lady who was just loved life. She was a primary school teacher, well regarded and well loved by all of her students. In fact, I had reason to find some of the sympathy cards recently, a few months back, and I reread them, which, I think anybody who has been in my position often, looks back at those times, and there were a number, more than several, literally tens and tens, dozens of cards from ex-students. So that was the type of lady she was. Love life, life of the party. Any excuse for a party, she'd be in it. She came from a very large family, many uncles and aunties, cousins, and she passed away in our home with, I think there was about 30 people in the home at the time. She came home from hospital. She spent some time in Prince of Wales, then went to the Walper Jewish Hospital in terms of palliative care, but she wanted to die at home. She was that sort of person. Loved life. She took a year off from her teaching degree at the time to actually be with our eldest daughter, who was having our first grandchild. So she was fortunate enough to have 20 months with our eldest grandson. We now have 6, and I know for a fact that she would have been just the most marvelous of grandmothers to our 6 children, 6 grandchildren who are missing her. So if that summarizes her, I think that does.
Thank you so much for sharing that. Pancreatic cancer often goes undiagnosed for a long time before it gets detected. Can you tell us a little bit about your wife's journey with the diagnosis?
Certainly. She was suffering from some of those very vague symptoms, stomach, toilet issues, the top bowel issues, and so on. sore back, but she had a sore back forever. But it wasn't a slightly different place. Best way of describing it was, as I say, March of 2013. In June of 2012, what was that, 8 or 9 months prior, we were actually traveling in France with a group of friends. And she always needed to be close to a toilet. She was suffering from things, but I now know 10 years, 11 years later, what I know of the signs and symptoms of pancreatic cancer. She had the cancer anything up to 12 months before diagnosis. This is a disease that's not detected early enough simply because the symptoms are so damn vague. You have a sore back, they send you to a chiropractor, they send you to a physiotherapist. You have insulin intolerance. They take the endocrinologist sort of picked it up. She was picked up by her gastroenterologist. She walked into the clinic. She was under his care because of the stomach issues, with a sort holding her back. Well-regarded gastroenterologist in Sydney said, I think I better investigate this. He couldn't actually do the endoscopy ultrasound. I'm going back 10 years. There wasn't too many gastro guys in Sydney that were capable of doing that. So he had to send us off to a guy who specialised in endoscopy ultrasounds, and he was the one that detected the cancer in March of 2013. As I say, I know now that she had the tumour anything up to 12 months prior to the diagnosis.
When we were chatting the other week, you mentioned that your wife said something quite controversial when she learnt of the diagnosis. Can you share with us what that was and what that time was like?
Yes, my brother's wife, our sister-in-law, back then, she's now a 25-year survivor, but back then she was a 15-year survivor of breast cancer. And can I preface my remarks by saying there's no such thing as a good cancer. My wife said to my sister-in-law, I wish I had breast cancer rather than pancreas cancer. The survival rate back then was about 90-odd% in breast cancer. In pancreas cancer, 7. It's now 13, baby steps. Breast cancer today is still in the low 90s, 92, 93, 94. I wish I had breast cancer. Horrible, very controversial, very provocative, but she said it to my sister-in-law.
Thank you so much for sharing that. Never think that to hear anybody say those words, but obviously a true reflection of just how incredibly dismal the current survival rates are for pancreatic cancer. To think that anyone who would have the experience that you've had would never want to come to face with pancreatic cancer ever again, hear of the word even ever again. But you chose a very different path and co-founded Purple Our World to raise awareness for the disease. Can you tell us a little bit about what inspired you?
Well, when Rochelle passed away, I spoke to David Goldstein, Professor David Goldstein, who was her oncologist. I just wanted to make some donations towards research or whatever. He told me about 4 organisations, Garvan being one. And can I simply say I wear a different hat as well. I sit on the steering committee of the World Pancreatic Cancer Coalition. Garvan is boxes above its weight. Australia, in fact, Australian researchers in pancreas cancer box above their weight. Garvan is world renowned in terms of this research that it's doing in pancreatic cancer, leading the world. So when my daughters came to me and said, we want to do a little bit more, I'd been in the not-for-profit space for some time and boards of educational and health and so on. But I said, I don't want to be a charity. There's only these 4 people here that are already doing some fundraising and funding of research, as minimal as it was. I said, we don't want to dilute the market. Don't want to dilute that fundraising. I want to go and help them fundraise. And one of my daughters is a media and communications graduate. She's a social media guru. we said we're going to raise awareness through social media. We're going to partner with the charities in that space, one of which was Garvan. So that's what we've done. We are simply a kitchen table with the family. We use social media to actually just raise awareness of this, for this neglected and forgotten cancer. Certainly 10 years ago, it was certainly neglected. Getting better, baby steps again. But really, back then it was forgotten and to a large extent neglected.
When you hear of the research that's happening, when you hear presentations like Marina and Anthony's presentations today, what excites you most, or what gives you the most hope about how the field has changed over the past 10 years or so?
Yeah, I've seen dramatic changes in the landscape over 10 years. I think one of the major observations that I had, I'm not technical, this is all gobbledygook to me to some extent, although Marina and Anthony have explained it very well, I think, in layman's terms. But I go to a few scientific meetings in this area and I sit there and be dazzled by a lot of this stuff. But what I have seen, in fact, let me just go back 10 years. There was no money. Young researchers were confronted with this very, very difficult cancer, very difficult disease in terms of detecting, in terms of treatments, and so on. So researchers weren't attracted to it. Researchers need to live. They need money to live on. They need funding. Fast forward 10 years, and I went to a scientific meeting last year, and in the room was 150 120 researchers, I don't know how many, the majority of which were under the age of 30 to 35. More money, more researchers, more research will lead to better survival rates.
That isn't a message of hope. I don't know what is. Thank you so much, Danny. On your mission for raising awareness, I guess what is the key thing that people take away from your story?
I think hope. We all must have hope. If I could just digress a little bit, got time. Pancreas cancer, all cancers actually, but especially in pancreas cancer, there are, I suppose, three things that we can do with our money. And it's a constant dilemma. I mean, the holy grail in any cancer is a cure. Well, more importantly, is early detection. Everybody gets a lump on the breast, they know what it is, they go and do something about it. There's screening for prostate cancer. What do we do about it? Men are going to die from prostate cancer before they get, so they'll survive it with it. That's one thing that we've got to do in pancreas cancer. The second thing is, once we detect it, we've got to treat it. Then there's a third issue with pancreas cancer, or with all cancers in a way, is providing a better quality of life and length of life once it is being detected. Limited funds. What do you do with the limited funds? Which one of these do you concentrate on? The true answer is you've got to do all three. You've got to try to get an early detection. You've got to try to get better treatments. And what we heard tonight or today, this morning, is in fact there is better treatments on their way. Getting it from the clinic into the bedside is extremely, from the bench to the bed is extremely important. Hope is what we need to do. There is baby steps happening. We'll get there. It's not something that will be in 5, 10, maybe it's a little longer. But we will get these survival rates up from that 13% currently in Australia. By the way, Australia is the highest. The US is lower. Europe is lower. UK is lower than that 13%. I think in some of the European countries, it's down into very low single figures.
Thank you so much, Daniel. Can we please give Daniel a hand? Thank you so much for sharing your story today. Thank you. A big thank you to you in the room and to those joining us online. You're all critical members of the Garvan family. Many of you are donors, partners for the future, and we couldn't be doing the work that we're doing without your support. So thank you so much.